Students, today the topic of my lecture is transcription in eukaryotes. In my previous lecture regarding transcription, I focused on transcription in prokaryotes. There is a lot of similarity between eukaryotic transcription and prokaryotic transcription, but eukaryotic transcription is little more complex than prokaryotic transcription. In eukaryotic transcription, different types of RNAs are being transcribed by their respective gene. As you are seeing in this picture, that is types of RNA, there are different types like mature RNA, mRNA, transfer RNA, tRNA, rRNA means ribosomal RNA, snRNA means small nuclear RNA, miRNA means micro RNA, siRNA means small interfering RNA. The last two, miRNA and small interfering RNA, are involved in gene silencing process. A process common in eukaryotes to silence the gene either by repressing transcription or translation. Gene silencing is common technique used in DNA technology to cure different type of mutated or diseased genes. Now, for transcribe the above mentioned RNAs, there are three different kinds of RNA polymerases which are present in nucleus, and we can call them as nuclear RNA polymerase. And on the basis of their sensitivity to alpha-amanitin, a fungal toxin produced by a mushroom known as Amanita phalloides, they can be classified and what type of products they form. RNA polymerase 1, which is located in nucleolus, transcribe 28S rRNA gene, 18S rRNA gene, 5S rRNA gene. It means it produces 28S rRNA, 18S rRNA and 5S rRNA. It is highly resistant to alpha-amanitin. RNA polymerase 2, which is the main polymerase enzyme which we will focus later on in this lecture, is located in nucleoplasm, and it transcribes all protein coding genes, means all type of mRNA genes, and thus it produces mRNA. It also produces snRNA like U1, U2 and U4. snRNA are designated as U1, U2, U4 because these RNAs are rich in uracil residues and they play an important role in processing of mature RNA, in particular splicing event, and microRNAs. And RNA polymerase 2 is found to be very sensitive towards alpha-amanitin. RNA polymerase 3, which is also located in nucleoplasm, it transcribes tRNA gene, 5S RNA gene and U6 type of snRNA gene. It shows moderate sensitivity towards alpha-amanitin. Despite or beside, we can say the nuclear RNA polymerase in eukaryotic cells, semi-autonomous organelles like chloroplast and mitochondria are also present, and they too contain their own genome. If they contain genome, means they carry genes, and these genes are also being transcribed. So the mitochondrial gene and chloroplast genes are transcribed by their respective RNA polymerase. These RNA polymerases are not RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. They simply are called as mitochondrial RNA polymerase or chloroplast RNA polymerase, and they transcribe all the genes present on mitochondrial genome or chloroplast genome, and they show resistance. They are very resistant to this alpha-amanitin. So now we will focus on RNA polymerase 2 because we will study the transcription of protein coding genes. RNA polymerase 2, as similar to RNA polymerase in prokaryote, is a multi-subunit enzymes. It contains more than 8 subunits. The major subunits are alpha subunits, beta prime subunits and omega subunit. Beta prime subunit is the largest and the main polymerase uh, enzyme or subunit. You can see the diagram of these subunits, 2 alpha, 1 beta prime and 1 omega, and you can see a carboxy terminal domain, also known as CTD domain, which is related to beta prime subunit, because these subunits are what? Proteins. So proteins possess N terminal domain and C terminal domain. Now it is found to be that CTD domain of beta prime subunit or RNA polymerase 2 is highly conserved. It possesses a heptapeptide repeat. Heptapeptide repeat means 7 amino acids linked together with 6 peptide bonds, and the repeat is tyrosine, serine, proline, threonine, serine, proline, serine. It can be designated as TSP, TSPS, and this heptapeptide repeat is found to be conserved in different organisms, even in plants as well as in animals. It means it play a vital role in transcription in eukaryotes. The repeat size may vary from 50 repeats to 60 repeats. It depend upon the particular species, and the repeat size is a signature mark of that particular species. Now, CTD, what important function CTD plays? It was found to be that CTD is phosphorylated during transcription elongation phase. We know that transcription can be divided as transcription initiation, transcription elongation, and transcription termination. So it has been seen that during the elongation phase, the CTD was found to be phosphorylated at particularly threonine and serine residues. Secondly, it also carries factors like these factors are involved in processing event. Factors like CPSF, cleavage and polynylation stimulating factor, CSTF, cleavage specificity factor, 3' for 3' cleavage and pollination, and it also carries a capping enzyme for 5' capping. Now you can see in this picture that the RNA polymerase transcribing a gene, that is DNA, and how you can see in picture that CTD is phosphorylated, and it is also carrying the factors like CSTF, CPSF and capping enzymes. Now we will move towards the regulatory sequence or the elements. The regulatory sequence or the regulatory elements are part of the DNA molecule or that particular gene which is going to be transcribed. These DNA sequences regulates transcription initiation, and it means these are the sequences which play important role in regulation of transcription initiation. Regulation means whether initiation of transcription must be permitted or not, or the initiation efficiency would be fast or it would be slow. So this is what called as regulation, and regulation of gene expression occurs mainly at transcription initiation level. It means these sequences play a very important role. In prokaryotes as well as in eukaryotes, the gene expression is mainly regulated at transcription initiation level. But in eukaryotes, there are various other levels also where the gene expression can be regulated, which we will study later on. Now these sequences or the elements, these are the DL elements, they may be present upstream or downstream. Upstream to transcription initiation site, that is +1 site, or downstream to transcription initiation site. They may be present far, up something around 1000 base pair from +1 site, both upstream or downstream. The common regulatory sequences are categorized as silencers and enhancers. Enhancers are those regulatory sequences or DNA sequences which increases transcription initiation efficiency, while silencer decreases the transcription initiation efficiency. Further, the consensus sequences or the elements, DNA elements, regulatory elements which are found to be just upstream to transcription initiation sites are called as promoters. In eukaryotes, the promoter contains -30, -75 and -90 type of consensus sequences. Consensus sequences, as I already mentioned you, these are the sequences which are highly conserved and present in various different type of promoters of the genes, of different kind of genes, and of which minus 30, if any promoter is containing minus 30, that is Tata box, means AT rich region. If any promoter containing minus 30 sequence, it means it is a strong promoter, while minus 75 is cat box, means the CAAT type of sequence, and minus 90 is a GC box. It is not necessary that all the three type of promoters are present in one single promoter. All these three sequences are present in one single promoter. In some promoter contains only minus 30, or they may contain minus 30 or minus 90, while some may contain minus 75 or minus 90. Now this is the structure of the promoter. You can see the plus one side, which is actual site of the beginning of the transcription. You can see minus 30 sequence, minus 75 sequence, and minus 90 sequence. Now one question arises, how the regulatory elements controls the transcription initiation. In this picture you can see the black shadowed portion is the regulatory sequences like enhancers or silencers. They may be a bending in the DNA molecule, and this is the RNA polymerase having CT domain. It is bounded over, it is covering minus 30 as well as transcription initiation site. So the enhancer silencer directly never interacts with the transcription initiation machinery. It interacts with the help of transcription factor, or we can say regulatory factors. These transcription factors or regulatory factors first bound to enhancers or silencers. These regulatory factors and transcription factors are actually proteins molecule. They bound to the enhancer or silencer by their DNA binding domain, then by their other domain, which can be called as activating domain if it is an enhancer. They may be called as a repressing domain if it is a silencer. By these, the activating or repressing domain, they interacts with the components of transcription initiation machinery, which is commonly known as transcription initiation complex, or we can say TIC complex. So by interacting with the TIC complex, these regulatory factors or transcription factors regulates the transcription initiation in eukaryotes. Now transcription in eukaryotes can be on the basis of its frequency. It can be categorized as a basal level of transcription or regulated transcription. Basal level of transcription means a normal level of transcription, and in this case, in such cases, regulatory elements or transcription factors are not required, because these are transcription of those genes which are called as housekeeping genes. And housekeeping genes are those genes which are expressed in all type of cells. For example, genes of histone genes. Histone genes are expressed in all type of cell, whether it is a liver cell, it is a muscle cell, it is a skeletal muscle cell, it is a cardiac muscle cell. So these histone proteins are synthesized in all type of cells, means there is a regular need of the synthesis of histone protein. Likewise, RNA genes, genes for some important biochemical reactions like glycolysis, they all are housekeeping genes, and they are always synthesized in their respective cells in a normal manner. While the regulatory transcription means this is the transcription of those genes which are under tight control, and they are expressed in some specific cells only. For example, insulin gene. Now, insulin gene is present in all type of body cells in an organism, but it is always found to be expressed only in beta cells of pancreas. So, when the insulin gene should be expressed and when not is controlled by such an enhancer or silencer or by transcription factors. Likewise, growth factors, growth factor genes are also tightly controlled, and their transcription is regulated transcription because they are involved in cell division. Now, formation of transcription initiation complex. So before we study transcription initiation in eukaryotes occurs when transcription initiation complex has been formed. And for transcription initiation complex, that is TIC complex, it requires RNA polymerase 2, the genes, all the substrates like NTPs, and all GTFs. Here GTFs means general transcription factors. Do not confuse with general transcription factors to the normal transcription factor or regulatory factors. General transcription factors are not involved in regulation. They are involved in synthesis of TIC, that is in transcription initiation complex only, in the formation of transcription initiation complex only. GTFs are therefore named as GTTF2D. Here I am not mentioning G before T, but you must know that TF2D, GTF2D, GTF2A, yeah, GTF2B, GTF2F, GTF2E, and GTF2H are the different kinds of uh, general transcription factors. So now we will begin transcription initiation proper. So first of all, you can see this is the DNA molecule, there is a Tata sequence, there is a +1 site. First factor to come is factor TF2D. Factor TF2D is further consists of two different type of proteins. One is called as Tata binding protein and another is called as Tata binding protein associated factor. First of all, Tata binding protein binds to the minor groove of DNA at particular Tata site. TBP is highly conserved protein and its DNA binding domain is highly conserved. It is rich in positive charged amino acid like lysine and arginine, so that it can easily bind to the DNA, negatively charged DNA, by the ionic interaction. So first it binds to the Tata site because it identifies the Tata site. Later TAFs binds to, that is TAFs binds to TBP, and the complete factor TF2D is synthesized. So when TBP binds, then TF binds, to complete TF2D is formed. Later on, factor TF2A arises and it stabilizes the binding of TF2D with Tata box. Once TF2A is bounded, it stabilizes the binding of TF2D. Now comes factor TF2F together with RNA polymerase. Factor TF2F act as a sigma factor, just like in prokaryotes. The factor TF2F recognizes factor TF2A and it recruits RNA polymerase to Tata site. So you can see the TF2F recruits the RNA polymerase. This is the CT domain. Note here that CTD is not phosphorylated yet, it is unphosphorylated. Now after binding of RNA polymerase 2, now comes factor 2E. It stabilizes the whole complex. When it binds to the complex, it stabilizes the whole complex. Then comes the most important factor, which is factor TF2H. So this is the last GTF which bind to the formation, bound to this initiation complex. And when factor TF2H binds, a complete transcription initiation complex is formed. Factor TF2D and factor TF2H are the most important factors in TIC. So when it binds, now what happens? Now TF2H has certain roles. It is responsible for helicase activity. So the helicase activity, that is the strand unwinding activities, is contained by possessed by factor TF2H. It also has it also hydrolyzes ATP, and it contains kinase activity for phosphorylating CTD domain. So it also behaves as a sort of kinase enzyme. Kinase enzymes are those enzymes which transfer phosphate group to their substrates. So here substrate is the CTD domain and it transfers phosphate group from ATP. So when the CTD domain is phosphorylated, it means the transcription initiation complex or the RNA polymerase is now ready to begin transcription, and that phase is called as transcription elongation. Now you can see in this case the CTD domain is phosphorylated, RNA polymerase has started elongation and synthesizing RNA. The GTFs are removed from their site and they can form another TIC complex to continue transcription by more than one RNA polymerases. Now as we have studied TIC, we also studied the structure of enhancer and silencer. So we will focus here a regulation of gene expression at transcription initiation level in eukaryotes. Transcription initiation level is the main level where gene expression is regulated in both prokaryotes and eukaryotes. As you know, and I mentioned earlier, the transcription factors are known as regulatory factors other than GTFs regulate synthesis of TIC by binding at regulatory sequences, enhancer or silencer. So here the regulatory factors, or we can say transcription factors, which are not GTFs, they binds to the enhancers and silencer and they control the formation of TIC. If they favor TIC formation, gene expression will occur. If they do not favor the formation of TIC, gene will not expressed. So this is the structure of a regulatory factor. You can see it is a protein having two domains. One is NH2 end, and you can see another is COH end. The NH2 end is called as N-terminal domain, and COH end is called as C-terminal domain. The N-terminal domain is known as DNA binding domain, and the C-terminal domain is known as activating or repressing domain. So this transcription factor by its DNA binding domain, which is NH2N, binds to either enhancer or silencer by weak interaction, and then by its activating or repressing domain it interferes with TIC formation, or it interacts with the member of TIC. Now you can see, first we will study DNA binding domain. The DNA binding domain of transcription factors are found to be highly conserved, and they are found to be rich in positive charge amino acids, so that they can easily bind to the negatively charged DNA molecules. They bind with multiple non-covalent interactions with their DNA. They can bind to both enhancers or silencer. In this case, you can see that in red color it is the transcription factor. The DNA binding domain, by their DNA binding domain, they are bounded to enhancer or silencer, and by their activating or repressing domain, which is COH, and they can interact with TIC formation. Now, DNA binding domain of various transcription factors have been studied extensively, and it was found that some common structures known as motifs, or they are also known as super secondary structures, are found in various different kind of transcription factors. So DNA binding domain is highly conserved, and the structure is common in different type of transcription factors. Some common type of structures are helix-turn helix, second zinc finger, and third one is leucine zipper, which is also known as dimerizing domain. In helix-turn helix, the NH2 domain contain two alpha helix. You can see the spiral type structure is an alpha helix, and it is rotating, it is turning to form another helical structure. So this is what called as alpha helix. So by this helix, they can easily bound to the major groove of the DNA molecule, and by their activating or repressing domain, they can interact with TIC formation. The common examples are the lactose repressor and the tryptophan repressor and homeo domain in Drosophila. The lac repressor and repressor are actually alpha helix. So in homeotic genes are expressed early during embryonic development. And homeotic genes are found in every organism, and they are first discovered in Drosophila. And during embryogenesis, that is development of embryo, they are the first genes to be expressed. They are responsible for the pattern formation in the developing embryo. If these genes are mutated, it leads to the formation of embryonic lethals. For example, they decide which part of the embryo will form head, which part of the embryo will form appendages, and other part. So in homeotic gene, it was found that 162 base pair of the region is highly conserved, which was called as homeobox. Actually, this homeobox, when translated, it gets translated, it from alpha helix, which is known as homeodomain in that polypeptide. That homeodomain is what? Alpha helix. So you can see in this diagram, in Drosophila, the body segmentation, the formation of different organs in their respective segments is all due to this homeotic gene. Second one is zinc finger. Zinc finger means these are finger-like projection, and they have zinc atom. They in their finger-like position projections. Zinc finger is of two type, C4 type and C2S2 type, where C2S2 type. In C4 type, there are 4 cysteine residues linked with the zinc. In C2S2 type, there are 2 cysteine and 2 histidine residues linked together with the zinc. They bound to the enhancer or silencer. By their activating repressing, they can interact with TIC. Likewise, leucine zipper are two different type of polypeptides, so they will form a dimer. So the leucine region is a non-polar amino acid, and by leucine they can easily with the help of hydrophobic bond, these two different proteins get dimerized, and their DNA binding domain interacts with the enhancers or silencer. Now, how activating or repressing domain works? It was found to be that activating and repressing domain in most cases are rich in acidic amino acid residues, and some are rich in proline and glutamine residues. So for first we are in first two pictures you can see how repressing domains works. So repressing domain means if it is repressor, if this transcription factor is acting as repressor, it first bind to the silencer by DNA binding domain, and it can bind to the Tata and by its repressing domain it binds to the Tata box. So the site is not available for factor 2D to bind. So when GTF 2D cannot bind, initiation process cannot, yeah, TIC formation never occurs. Suppose TF2D has bind and all factors and RNA polymerase is recruited here, but by repressing domain it binds to a site known as where factor TF, where factor GTF2H is supposed to bind. So when 2H is will not bind to TIC, it cannot phosphate CTD, elongation will never occur. So in this both cases there is gene silencing. In activating domain, in the transcription factor bind to an answer, and by its activating domain it can bind to a silencer site, so that repressor cannot bind. When repressor will not bind, the TIC it will favor the formation of TIC and transcription. Now elongation process is same as of prokaryotes. The RNA polymerase will move towards the 3' end of the DNA, of the template DNA. Now we will move towards transcription termination. Transcription termination is different in case of eukaryote as compared to prokaryote. But before transcription termination occur, some modifications occurs in the precursor RNA, and these modifications are a part of actually RNA processing. The first one is 5' capping, then comes the 3' cleavage and polynation, and 3' cleavage and polynation is also known as transcription termination, and after transcription termination splicing occur. So in this picture you can see the RNA polymerase is transcribing, and in red color it is the newly synthesizing RNA. You can see CT domain having factors, and it is present in phosphated form. So the CT domain contains capping enzyme. The capping enzyme is responsible for formation of 5' cap at the 5' end of the newly synthesizing RNA molecule or pre-RNA molecule. It is necessary because it protects RNA from exonuclease activity. It is also required for correct orientation of ribosomes during translation, because RNA has to travel in eukaryotes. RNA is not rapidly translated. After being transcribed, it has to cross a nucleus, and it went to cytoplasm where it can be translated. So there is always a threat of being digestion by the nuclease enzyme. So both ends, the 3' end as well as the 5' ends, must be protected from the exonuclease activity. So capping is must. Now what is capping? Capping is addition of 7-methylguanosine. You can see the guanosine which is methylated 7,5%, and by 5' to 5' unusable triphosphodiester bond, it is attached to the 5' end of the RNA. You can see the adenine, and then it is a uracil. Further, first two bases of the RNA are also methylated at the 2' OH end. So this protect the 5' end. Now comes cleavage and polynation. Cleavage, which is also known as, and pollination is also known as transcription termination. So when RNA polymerase is transcribing the gene, you can see in red color this is a precursor RNA. When sequence like AAU, AA, and later on GU-rich sequences arises on RNA, these sequences are called as cleavage and polynation signal. And when these sequences arises, CTT domain recruits CPSF at AAU-AA region, and CSTF is recruited at GU-rich region. And many cleavage factors then cuts or breaks the phosphodiester bond between AAU-AA and GU-rich region. So when they cut the RNA is detached from the transcription machinery. Now this particular 3' end is free, where poly-A polymerase enzymes is ready for polyadenylation at 3' residue. It forms a poly-A tail. Poly-A tail means a tail containing of adenine residue. It may be something around 250 nucleotides. So this poly A tail protects the RNA from exonuclease activity from 3' end, and the 5' cap protects the RNA from exonuclease activity from 5' side. Now this is the RNA which is ready for splicing so that it can form mature RNA, but still transcription is continuing. A part of our RNA which is present, which is still being transcribed, is transcribing. Although this mRNA will not be part of the this RNA will not be part of mRNA, but how termination occurs, there are two different views of transcription termination. The first view suggests that when RNA polymerase is still transcribing after 3' cleavage and polynylation, RNA polymerase senses absence of 5' cap at GU rich region. And though you know that all the cleavage factors and all the capping enzyme factors, they all are recruited by CTD domain earlier. So there is no capping enzyme present at CTD domain. So this is sensed by the RNA polymerase and the subunits gets detached or disassociated. In second view, it says that since there is no CPSF and CSTF because they are recruited earlier, so the absence of CPSF and CSTF is sensed by the RNA polymerase, and it leads to the dissociation of the whole RNA polymerase, and it leads to transcription termination. Thank you.